In this video, we're going to talk about transport of molecules across the membrane. So in a previous video, we already talked about which molecules can get through the membrane easily on their own, and these would be the nonpolar molecules, especially small nonpolar, but even large nonpolar can get through easily, and then sometimes small polar molecules like water can get through, but relatively slowly. So when they're moving across the membrane on their own, for that we would be talking about the passive diffusion. So passive diffusion basically just means that these molecules are able to get across without any help. They pass through the nonpolar hydrophobic interior of the phospholipid bilayer, and that is diffusion because things are always moving from high to low. And it is passive because it's moving there across that on their own. And again, the reason that these molecules can get through across on their own is because they're not repelled by the hydrophobic interior of the phospholipid bilayer if they're nonpolar, therefore they make it through. However, polar molecules, especially larger polar molecules and ions, anything with a charge, they need help to get across the membrane. So this is going to be called facilitated diffusion. And again, it's still diffusion because these molecules are moving from high to low, but now they need help. So this might be in the form of a channel. And the channel basically creates an aqueous pore through the center that is perfectly situated for sodium ions, potassium ions, or anything that it's moving across, including water, if it's an aquaporin. And then we have the transporters. Transporters are much slower because they have to actually bind to the substance, and they constantly are changing conformation from the inside to the outside. So basically they're constantly going back and forth, opening to the outside and then opening to the inside. So this would basically look like that, bind, and then it would open on the inside as you see here. Now when things are moving from low to high, when they're moving from low to high, for example, this molecule right here that has low levels on the outside and it's moving in to where there are higher levels on the inside of the cell, this is going to require energy. And this energy often can come in the form of ATP, but there are other forms of energy as well. And this is active transport, when anything's moving from low to high and therefore requires energy. Now sometimes that energy comes directly from ATP, as we see here. Other times that energy can come from what's called coupled transport. So down here we can see that initially ATP is being used to pump hydrogen ions from low to high. So the cell is pumping them using energy to move them from low to high outside of the cell. And it's doing this in such a way that it makes a gradient. So gradients are very important to cells. So cells need to have high concentration of certain ions and certain molecules on one side of the cell and lower on the other. And in order to do that, it has to use active transport to pump ions against the gradient. Now later on, the cell can use this to do work. So when those hydrogen ions are moved through facilitated diffusion, are moved from high to low, we know that that gives off energy. Anything moving from high to low gives off energy. And that energy can be coupled or used to do work. And in this case, that work is move things like sucrose from low to high. So the cell wants a high levels of sucrose inside it so it can digest and make ATP. And also it needs high levels of glucose, things like that. These work very similarly. So that movement of sucrose from low to high takes energy, and it's done by moving the hydrogen ions from high to low at the same time. So this coupled transport basically moves one molecule from high to low to power another moving from low to high. And this ultimately is that coupled transport that we just talked about, and that allows work to be done. Now, if this was the end of the story, eventually pulling the sucrose in would also use all of the hydrogen gradient, and we would end up with an even amount of hydrogen on both sides. So in order to keep doing this, the cell has to spend ATP energy to pump the hydrogen ions back out so that this cycle can continue. As we said, active transport can use ATP to move something from low to high. So in this case, it's moving hydrogen ions from low to high. We can see that we have a high abundance of them on the outside of the cell here, and we have a relatively lower abundance on the inside. So if we're moving them out, we're creating a gradient of hydrogen. And this is important. This is how the cell creates gradients all the time. In fact, the cell will create them such that it will be able to use that gradient for other purposes later on. For example, it might use the energy of hydrogen flowing from high to low, because we know anytime something moves from high to low, that gives off energy. And in this case, this is coupled transport. The energy of hydrogen moving from high to low in this direction is being used to pull sucrose, in this case, from low to high. So the cell needs a high levels of sucrose inside, just like it does glucose, and so it's going to pull them inside so it can digest those. So it's taking it from the bloodstream where there's lower amounts, pulling it into the cell where there's higher amounts. That takes energy, and it does that by moving hydrogen from high to low to pull glucose from low to high. So the movement of one thing, so one thing moves from high to low to power another movement from low to high. But again, if this kept happening, eventually all of our hydrogen is going to become inside, and we're going to have an even hydrogen gradient. So we need to continue to use ATP to pump the hydrogen back out so that this can continue to happen. We continue to have a cycle across the way.